right, thank you so much, Hernan, for the introduction. Yeah, I think that there's a lot of students and postdocs. I think one thing especially in these sort of uncertain times, it's important to take risks and figure out what is really missing. So you can, I feel like everybody should have a little stubborn view of their world of like, oh, these people, these established scientists are not doing things right. I need to do things differently. How can I do things differently, right? So if you forget everything else in my talk, maybe just remember that, please. Cool, and I'm just gonna plug one other thing that I'm involved with, which is launching a journal in biological physics called PRX Life. For the next several months, APC fees are still waived, so because it's a pretty new journal. So if you have great research at the interface of physics, engineering, and biology, please consider the journal for submitting. Okay, so basically throughout my scientific career, I've been really interested in understanding how shape and dynamics is maintained in biological materials, right? So this is a field, if we think about it from physical matter, the fields of material science, condensed matter physics, fluid dynamics, right? And it's a very well-established discipline that has made great progress in understanding how to engineer physical matter, right? This is what gives us silicon chips and technology, right? All modern forms of materials, right, is based on our fundamental understanding of how fluids flow, how solids deform, what controls electron transport, right? And we did this by looking at geology, rocks, our external world, but then trying to take that matter and start manipulating it in the lab and do more and more experiments, right? And if you look at shape and dynamics over time, you can ask, what's the difference between a drop of water splashing? This involves a fluid with a surface tension and a lot of different scales, right? At the smallest scales, it's actually a singularity that determines where drops break off, right? And compare that to something that we see under the microscope of Drosophila morphogenesis, right? All of these things undergo dynamics where the mechanics of the material is really allowing for the evolution of shape over time. Right, so if we think about fields of condensed matter, right, it's a bunch of molecules, and these molecules come together with strong interactions to build something that at larger mesoscopic scales can be characterized with order parameters that then determine how external forces then control the emergent dynamics. Another thing I like to say is that we discovered Hooke's law and fluid dynamics way before we had possibility to, in the case of Hooke's law, we had any ideas of what atomic theory was. We didn't even know atoms existed, right? So we only had a mesoscale description of it, right? And in the case, even after you knew of atoms, there was no way we could simulate them, right? So all these models, these coarse-grained emergent approximations of physical matter was done with no understanding or no ability to capture simulations of all molecules and their interactions at these scale, right? This is the strategy that physics has used to understand emergent phenomenon and has been very successful in physical matter, and we hope in living matter that we can do the same type of thing, right? Now, the problem is as soon as you start thinking about matter that's built out of biological molecules, first of all, there's a much bigger diversity of interactions and shapes and of the biological molecules. They're strongly driven out of thermodynamic equilibrium because of the external energy, right? And then there's going to be new types of order parameters and ways of describing the biological and living matter, right? But perhaps even more importantly, there's all these feedbacks that exist, right? So there's feedbacks between how external forces change the state of the biological molecule and then impact the local material properties, right? So it becomes a much more complicated problem that requires new frameworks about how we study emergent properties of strongly interacting molecules, right? And so just listing in words here, they're strongly driven, right? It's far from equilibrium. There's lots of internal stresses driven because of internal activity. And there's all these feedbacks that are happening, right? So how do we actually tackle this? And I think what I'm going to hope today is tell you about what we are at in terms of the modeling and maybe a path forward in my little version, my little sphere of this. Okay, another important thing, I mean, there's people coming here from all different scales, from the subcellular up to the tissue scale, is that when we think of biological matter, we can think about a coarse grain approximation at any of these scales, right? We could think about this happening as just collections, simple collections of macromolecules within the cell, but then also think at the tissue scale of lots of collections of different types of cells, right? These span vast length scales and time scales, and the feedbacks go from the molecular, where you can actually engineer life, right? That's how you actually control what the composition is, to this emergent tissue scale phenomenon, right? So you have to think about at each of these scales, from molecular to the subcellular to even cellular scale, where there's state transitions of individual cells, up to the tissue, of how you're going to tackle this problem, right? But each of these scales still has these features, right? Where there's all these different components, there's all these different scales and feedbacks that spanning these scales. So it becomes a really complicated problem. All right, and so how do we even think about building predictive frameworks for this? Okay, so first, this question of coarse graining, right? Like, one is you could take a problem at any given scale. It has a bunch of collection of macromolecules, some chemical feedback loops, and we want to describe this bit of complication of molecules into like a blob that can be characterized, say, an elastic modulus, a viscosity, maybe an active stress term, right? So this has been, I think, one of the challenges and successes of physical biology, biological physics over the past 25 years or so, right? Is really trying to think about what are the kinds of order parameters and what kind of new order parameters do we need to describe. And this is a field known as active matter physics, where the insight was that a really important new order parameter to add is the notion that there's internal stresses driven within active matter, so you need to add an active stress that allows for spontaneous flows and shape change and internal stress buildup. So that over the past 20 years has been built up and is now an established subfield of condensed matter. One thing that's newer is really, now you take something that's active that has internal stresses, and now you ask, in biology often these parameters change over time, right? So the elastic modulus might evolve spatially and temporally as an object is changing shape, right? This, I would say, is not quite an established field of physics. It's something that's emerging now, known as adaptive matter. Really, what are the rules that there could be transitions in properties that are happening, and how do these happen smooth enough, right? One of the things that, if you take movies of natural systems, natural living systems, right, they evolve smoothly across scale of length and time, right? They never just rip themselves apart, which is sort of surprising given the amount of internal stresses they have. So what is the nature of these transitions that can occur to allow for these smooth transitions across all these scales? So this is something that I would say is not established yet. There is no protocol for me as an experimentalist to say, I want to probe the adaptive nature of this thing I'm studying and understand the basic premise of it. And even less understood is really thinking about this bit of matter as an information processing unit, right? So instead of thinking about it as an object that is like a physical object, I want to think about how it takes inputs of external stimuli or internal stimuli to then do physiology, right? So this is, brains, this era of AI, right? Everybody likes to think about information processing. Neuronal cells are one class of cells, right? You could think of all cellular networks or even networks of macromolecules as bits of matter that can then also process information to then modify its own state. And the emergence of all this is physiology, right? This is not, as far as I can tell, there is no protocol outside of our notions of neuroscience about how do you actually take a measurement that I'm doing to ask how many bits of information is it storing to be able to do something useful, okay? So this is what I think is really exciting going in the future about where the boundaries of understanding the physics of life, of living matter is going. Okay. So I'm going to talk about these questions and sort of the evolution of these types of model building in the context of one simple system that we've studied a lot in my group, which is just really understanding the systems that are underlying a single cell, a single adherent cell. So this is a fibroblast cell that's spread on a two-dimensional surface. You could also think of this cell as migrating through your body, and it's important for building and remodeling tissue, the extracellular matrix. Right, and what we're looking at here is the actin cytoskeleton, and we spent a lot of time thinking about the material that builds up the actin-myosin cytoskeleton. So here, myosin-2 mechanoenzymes generate internal stresses and transmit those stresses across the cell that then are transmitted through cell matrix adhesions or focal adhesions to the external environment. And this allows the cell to generate contractile tension and force on its external matrix. And there's feedbacks between the matrix mechanics, how many adhesions there are, and the amount of contractile force, right, that are occurring. Now, this is sort of a snapshot cartoon if we watch these processes live. And again, these are movies that were enabled by, development of fluorescent probes 20 years ago, imaging technologies that are 20 years old. These movies, the ability to take these kind of data is not new, but we're still having challenges of how to understand this data and how to interpret it, okay? But this is where we were at, like when I started my research group. We can take movies of myosin-2 molecules changing, and you watch all these networks that can remodel as a cell is smoothly moving forward. Or you can tag the focal adhesions and look at the distribution of the adhesions as the cell is moving, and at the same time put the cell on a calibrated elastic substrate and directly measure the physical forces that the cell is exerting on the underlying substrate, right? And so the questions, right, are still the same, is like, what regulates? How do you quantitatively and predictively understand how the magnitude and the distribution of applied stresses is regulated across the cell? And how do these structures, right, these actin-myosin bundles are of strong internal tension, how do they maintain smooth transitions to be able to remodel and assemble and disassemble and maintain tensional homeostasis despite all these internal stresses? Okay, so with this kind of data that modern, quantitative biology and biotechnology enabled, right, you think about the very standard way that we've always done model building, which is this feedback loop between making quantitative measurements and manipulating the cells through engineering or perturbation experiment, and then working to build an interpretable model that's grounded in understanding of physics and chemistry through the lens of mathematics, right? And as I mentioned, right, it's really been the progress made in both cell engineering and biotechnology and imaging that has allowed us to now get large quantities of data to be able to study these processes. So it's been a great success. And another way of saying this is like, again, about 25 years ago, we were able to know most of the molecules, right? And it's been a question of how all these molecules together are really working to build an emergent machine. Okay, so in this case, a success has been that you can make coarse-grained descriptions of something simple as an adherent cell. So, for instance, you take this cell, there's all these dynamics going on between actin-myosin. We had showed, and others have shown, that like you can take these measurements, you can do a coarse-grained description of an adherent cell as an elastic sheet with uniform adhesion to the substrate, and that has internal stress. And so all you have to do is describe the cell with a parameter that measures a internal stress and a line tension, and you can get measurements, or you can make a prediction of what your experimental force is at, which is the middle data, with this very simplistic physical model, right? So this would say, I'm coarse-graining all that actin-myosin and the adhesions as a pre-stressed elastic sheet. Okay, so that's great that we can do that, and this is just one small example, but what are we currently missing, right? We don't have any time prediction here, right? You can sometimes in very simple case, make a prediction in time, but it's very limited in that. It doesn't capture anything about the underlying mechanism of those parameters, right? Or how it might sense environmental stimuli. You can just have a description. It doesn't have any really of the stuff that we think of as mechanobiology, of really how the molecules are doing force sensing and response, or how these parameters change in time, and it really is limited in its ability to capture spatial variation. So one thing that's happened more recently, again with bioengineering tools, is the ability to now do local perturbation experiments to measure force and spatial and time-dependent responses to the cytoskeletal networks, right? And this is a technique that was developed, brought into my group by Patrick Oakes, and Kate Kavanaugh was the second generation student, and she's in the audience now. that really allowed us to take light and then manipulate locally and use it like as a little tweezer inside the cytoskeleton. So in this case, the tool that we have is that a light-induced activation of a GEF, a guanine exchange factor for Rho A, that then activates actomyosin to basically apply a local contractile tension. And so these are two movies that Patrick took where you're just activating Rho, and you can see both the traction stresses can be dynamically modulated, and then you can drive and reorganize the actomyosin flows. Now, why this is so important is before, all we were looking at is the natural state, the natural dynamics. So we weren't able to do a perturbation response experiment, right? So optogenetics is a way that you can start to do controlled perturbation response experiments that allows us to sort of unravel the logic by which these materials are making decisions. And so an example of this is we're able to use this optogenetic thing to apply a local contractile tension to a stress fiber, watch how it flowed towards a place of high stress, and then watch what happens when you take off that additional stress. And what we observed is that those actomyosin bundles are basically behaving like elastic bands, right? The response that we see is largely an elastic-like response. That is, you apply a force, it deforms, you take off that force, it goes back. Right? And so we could model this like elasticity, but we can then now see that that elastic response requires certain molecules, okay? So this is an example where we have eliminated a molecule that I'm going to describe later called Zixin, where you now activate the local contractile stress. The contractile stress is exactly the same here. The only difference is now is that when you activate the stress, everything flows towards the center and doesn't recoil. So it's behaving like a viscous fluid. Right? So now what's happening is that now if you think of this model as a spring and an active stress and a dashpot of viscous in series, that viscous relaxation time is now going to be predominantly fluid. All right. So now we haven't changed anything about how the cell is generating contraction or how the contractile force is transmitted across the cell. All we've changed is a mechanical response, which is only seen by if we do this local perturbation. Now, this molecule that I told you was so important is actually something that we and others had studied a lot, which is involved in a feedback loop to sense to how to repair actomyosin stress fibers under enhanced tension. So this molecule Zixin here has a series of zinc finger domains called limb domains on one side, and then other region of the molecule has binding sites to the actin crosslinker alpha-actinin and VAS, so then when you bring that molecule to, you increase the concentration, it nucleates lots of actin cross-linking and actin filaments. But this limb domain area is important for sensing where actin filaments are under tension. And if that is required for repairing a stress fiber when it's damaged by increased mechanical stress. Okay? So what this means is that when we measure something that's like an elastic response, it's not measuring like stretch of a spring. It's actually showing you that the origins of it is from a force-activated feedback that's mediated by the Zixin. So the idea we have is that there's a mechanical response of the stress fiber, but then because if there's any external force perturbations that are applied to that stress fiber, it initiates this feedback, which happens pretty rapidly, that's mediated through Zixin, to remodel the actin to facilitate something that's an elastic-like, right? So anything that, even something that's simple, like behaving like an elastic spring, is mediated by a force-activated feedback. This was really interesting to us and others because these limb domain proteins do not bind to actin filaments when there's no tension on the actin filament. They have very weak binding affinity. But if you put the actin filament under tension or compression, they have a higher affinity to that segment. We have reconstituted this process by purifying actin filaments, purifying these limb domains, and watching the process emerge. We and Greg Alushin's group at Rockefeller have been working to reconstitute this, and we think this is showing us a new force-activated binding site of actin. And indeed, if you do molecular dynamic simulations, which were calibrated in Greg Voth's group, his group has worked for over a decade to build a really nice multi-scale model of actin filaments and being able to apply different amounts of force to it. What our student, Bill, showed that is as you apply force to that actin filament, there is like a metastable configuration of that actin filament that opens up and exposes a binding site along the actin filament to which we see that limb domain preferentially goes. So it's a binding site that's not available under normal situations. It's only available under tension. Okay. Okay, so this is an example where you have now a polymer. When it applies a force, you apply force on that polymer, in this case actin, a new binding site's exposed, this limb protein then binds to that binding site. And so we think of these limb proteins as a new class of basically of potential force sensors that are mediating these force-sensitive feedbacks in adherent cells. Now, the cool thing about these limb proteins is a previous postdoc, John Winkelman, when he was in the group, noticed that the whole family of limb proteins, there's only 4 in fission yeast, so a really simple model organism that has only one contractile organelle, the cytokinetic furrow. But this family just exploded at the time when you started building multicellular tissue. Right, and so there's over 30 different limb proteins, and I think 6 different classes. And they often localize to different force-bearing adhesions in the actin cytoskeleton, either places where cell-cell adhesions are, cell matrix adhesions, or the actin cytoskeleton. We wanted to see how ubiquitous this force sensing was, whether it was only Zixin was a force sensor, or these other limb proteins also had force sensing. And so what John Winkelman did was do an assay where he expressed in a cell line that had GFP Zixin in it, so a known force-sensitive process, he co-expressed constructs of the limb domains from 30 different limb domain proteins and asked what was the amount of recruitment of these different limb domains to the places where Zixin was exhibiting a force-sensitive recruitment. All right, and so what he found here was that there were over 18 different limb domains that were exhibiting similar but variable amounts of force sensitivity to places of stress fiber strain. Right, so this is not a singular feature of the limb domains of Zixin. It's really it's a larger class of force sensors that are happening within this whole limb family of protein. So we're really excited now to explore how these are functioning mostly in multicellular tissue. I don't have time to go into it entirely, but a postdoc, Shailaja Sitharaman, who's going to be starting her lab at the MBI, has shown that another limb domain protein, which only consists of 4.5 limb domains, that's the name 4 and FHL2, is a mechanostat that actually regulates epithelial physiology. So I'm just going to summarize what she found here. So this is another protein that exists in the cytoplasm and then binds to actin when the actin is under increased tension. And instead of feeding back to the stress fiber mechanics like we think Zixin did, so that had a feedback loop here, what FHL2 do is actually activating Rho A. So this is a positive feedback loop to a place where there's applied stress. It localizes to the actin filaments and then initiates a positive feedback to actually increase the amount of contractile tension. So it's setting the contractile set point by which adherent cells are actually modulating their increase in tension. More interestingly is that we have that FHL2 is not just regulated through its force-dependent binding to actin, but it's also transcriptionally regulated. So under places where there's high increased stress, it's transcriptionally upregulated to increase more FHL2 expression. Right, so there's now 2 different timescales. One that's associated with cytoskeletal mechanobiology, but now a slower timescale to understand how this feedback is altering the cell state. And again, so what Shailaja has shown is that this kind of force-sensitive feedback is operating to allow a transition in endothelial cells to go between places that are associated with low inflammation and low permeability in the vasculature to places of high permeability where the cells, there's lots of gaps between the cells. This transition in permeability can be modulated by external or internal changes to contractility, so changes in the type of flow that you apply to the cells can modify the mechanical state of the cells. Changes in internal row activity can also modify this. But what Shailesh showed is that each of these then also feed back to FHL2 expression. So if you suppress the expression of FHL2, you stop this transition between the sort of system between a low and high permeability state. Okay. All right, so what I've got through so far is a sort of a foray into sort of how we've approached these problems, right? On one hand, we've been trying to build coarse-grained descriptions of the systems. At the same time, we're learning more of the mechanochemical systems that are giving rise to the physical properties of these adherent cells. And we're not the only people who take this approach. Like, this type of approach has been done in all sorts of areas of cellular and tissue scale biophysics, right? And I would say where we're at as a field is like we have these sort of qualitative descriptions of a systems level, and then we have, coarse-grained models, right? And what are we going to do about this? And as I said, just to reiterate what I said at the beginning, this is really challenging for model building, right? There's all these different length and time scales. We're never going to know all the mechanochemical feedbacks, right? How are we going to actually make a useful model if we can't measure everything that we think is important? And I would argue that we're never going to be able to measure that. Like, even despite all of our attempts to measure everything at once, it's always going to be imperfect, right? And so what could we do? And so this has come to really think about that we need to, and this has been a challenge in the field, is that we really need new approaches for dimensionality reduction for mechanobiology or just living matter, right? And so, in the examples of the kinds of models that we can build, right, this is the microscopic or agent-based model. This goes with the worldview that if I measure everything, I will build a really useful model, right? But these models often break down pretty quickly because there's just too many agents and agent-based rules. And again, I think it's impossible to measure everything accurately enough to be able to have a useful description, right? And then on the other side is continuum physics-based theory, which is this like glorified, beautiful view of what physics modeling should do, but it really lacks a lot of the essential biology that we think is important to living matter, right? And it also lacks predictive power of forward dynamics or physiology, right? So the challenge, so these are two examples of stuff that we've done with theory collaborators over the past 10 years. The challenge is really how to properly reduce the dimensions. And for students in the room, and I guess, us older people, the way you do is you measure something, you're like, I have an idea of what's important, let's make a mathematical model about this, and so there's this feedback, right? But we never had a good procedure to use for dimensionality reduction. So we've been trying to develop new ways of doing this using AI. And so the last part of my talk, I'm just going to talk about a more recent collaboration with Vincenzo Vitelli's group. So this work was really led by two students in his group, Matthew Schmidt and Jonathan Colin, who were developing AI tools based on data that was collected in my group by Shailesh Raman and John Devani, and then Stefano Sala and Patrick Oaks' lab. So it was a really wonderful, really started in the pandemic driven collaboration to see how do we use AI in our mechanobiology model building. Okay, so here, the first question is, could we do the procedure in current modern day neural networks, are they useful for telling us what information is sufficient to make a prediction about a physical field? All right, so in this first step, we're just going to ask the question, if I have my image of my focal adhesion protein, and again, I'm going to use Zixin because now you know it's a force-sensitive focal adhesion protein, right? So we think it's going to work, and this has been my strategy with this tool development, is use something that you have a very good intuition about first, so you know when it's bullshitting you. All right, so you start with your images of Zixin. You have your measurements, experimental measurements of the traction forces, how much mechanical tension the cells are generating on the substrate. And now you train it in using a convolutional neural network. You ask, can I train a convolutional neural network to make a prediction to accurately predict the traction forces? And so how well is this predicted field going to do if I minimize the loss of that trained neural network? And the answer is it does a pretty good job, right? Like it was shocked. We were able to train it with a data set that could be collected from a student in an afternoon's worth of data, right? So very easy amounts of data for an individual lab to collect with imaging. Right, and I guess as a rule of thumb of how much error is there, right, when we had a whole test set of images that were used to, or training set of images used to train the neural network, then we had a whole test set of images that had never been seen by the neural network using different cells with different geometries, and the noise that we got from the test and the training set were no different from each other. But even more surprising is that we trained this on wild type conditions where the cells were in control with a high amounts of contractile stress. And then what we did was we fed into the neural network a cell where we had applied a very strong perturbation where we relaxed the contractile tension by applying a rho kinase inhibitor. And the neural network still did a very good job, right? This means that it's predicting, making a prediction of the cell traction force way outside the regime at which it was trained in. And what this means is that, you know, oh, I didn't actually play the movie. Let me see if I can play it again. Maybe it just didn't loop. What this means is that all the information in that control image was sufficient to make a prediction of what happened under the strong perturbation. And this is a hypothesis that I have that, you know, that I don't know, we'll see how it pans out. But like, part of the problem with model building of especially tissue culture cells is that every cell is different, right? This is why people have used historically model organisms and developmental pathways, because everything is so canalized and robust. Now, the thing is that when that variation, especially in tissue culture cells, now becomes a feature and not a bug for model building when you're using AI. And actually, it becomes a big problem when you're trying to train neural networks on all these developmental processes because there's so little variation. So I think that all this variation that we've been trying to avoid is now becomes a useful tool to explore and really quantify the environmental landscape that these cells are operating in, right? Okay, so we're, and I also want to just emphasize that, you know, this was our first foray into doing this, of really seeing where we could use this tool for us as experimentalists who want to understand basic biophysics, right? So a lot of this stuff is just new for the first time, and I think will require technique development further. But one question you can just ask simply is like, what was important about the field of Zixin? What was it in that image that actually gave you useful information about the traction stresses? Well, the first thing, you can just take out all the features of the protein distributions and just ask how well does the cell mask do? And it does a pretty good job. Which, that's really little amounts of information. These are just zeros and ones, right, in this image. All it's giving you about is cell geometry. And I guess this could be surprising, except I already told you that one of the coarse-grained models we used only was based on the cell geometry, right? There's this nice 2-parameter coarse-grained model. So in hindsight, it's not that surprising that that works so well. But obviously, the distribution of the proteins is giving you a little bit better of noise floor, right? But then if you put in something that's, you know, not really connected to the force field like mitochondria, right, you get really much even worse noise, because now there's information in that image that is not useful at all in terms of the force generation, right? So this is a tool that can start helping us taking imaging data as a procedure to say, is this information I'm collecting useful about the thing I care about, right? And so so we can just ask that for all the different types of data that we collect. It's like, how is this field better than this other field? Is the data I take at this time point predictive of the next time point? The opportunities are endless of really using this as a way to know, is what I'm measuring useful of the thing I want to predict? Okay. Once you have a trained neural network, right, this is like the most baby model version of a virtual cell, right? I know it's a big thing right now, but this is like a synthetic model to be able to make predictions about what your cell would do. In this case, it's a simulator of what happens if I give it an image distribution of intensities and distributions that are modeling where Zixin is located, and makes a prediction of what the force field would look like. Right, so now I can run through in this synthetic data set all these parameters that would take an experimentalist a lot of time to actually collect at this point, and sometimes wouldn't happen because, you know, biology is limited on what cells will actually do for you. To explore a whole field of like asking, under what conditions of adhesion size and orientation and distance from the edge will you actually maximize the force that is output at that adhesion site? And what comes out of this is super cool. You can see this peaks, the amount of force peaks sort of right around here. And from 10 years of 10 different labs studying focal adhesion mechanobiology, that's exactly what all those labs learned too, right? So it's picking out a length scale and intensity of where adhesion sites are under the most amount of tension, stress that you can now do in this simple simulation. And I would argue that then you want to go and test that again. But now it's also a procedure to learn quantitatively these force-sensitive feedbacks, right? So this is an example where now we can quantitatively ask for an adhesion of a certain size, right? If I change the Zixin intensity at that adhesion at different lengths, what is the force-sensitive feedback that I see? And what you see here is that it actually varies. There's lots of different parameters. So it varies on both the adhesion size, its location, and only becomes force sensitive under certain conditions. And this is an example of how there's so many different parameters that just looking at the natural condition, these simulation approaches can start to learn the force sensitivities that would be impossible to distill out otherwise. Okay, so now I'm just going to step back again. So we have our experimental data. I should just say, we want to get to here. We want to have some interpretable theory that we want to get to that makes us, have us this grand unified physics of living systems that are able for forward prediction, right? That will also allow for engineering, right? These neural network models are very accurate representations, at least for the systems we've studied, of the data set itself, but it's completely uninterpretable, right? We have no idea why it's giving us a good result. We just know it is, okay? So it's a good model, but it's not interpretable. It's not really what we want in the end. And there's this big gap in between these neural network models and sort of an interpretable physics-based model, right? So we want procedures and tools to make these neural networks more physically interpretable, but also to augment physics that we can have with more biochemical realities, right? And you could put any kind of biology that you want in there, whatever biological reality. So I'm just going to briefly describe these two, what we've been working on to address both of these two arrows. So one is an example where now you have the ability in a neural network to, you have a neural network that works, right? So we have a network that is trained to predict forces from an image field. And now the question is, could I learn a mathematical basis set of equations that is sufficient to describe the data, right? So this is really just learning the Green's functions that are necessary to go from my images of Zixin to a vector field of the traction forces, right? And so this is, what we're doing here is embedding a Green's function layer. We hand it a basis set of mathematics, and then we learn the Green's functions of the images that are necessary to make a prediction of the traction force field. And what we learn from this is that it can be safely described by two mathematical expressions. These are your Green's functions. You can see that the yellow curve has a longer length scale that sort of smells like elasticity, right? And the pink curve is very well localized, right? And just with these two functions and the source fields of where those Green's functions are localized, you can make a prediction of the spatial distribution of forces. Now, it's like, this is now a very reduced in terms of dimensionality of the description of the forces, and it gives you precise mathematics. It has no physics in it, but it's giving you the necessary mathematics, which I think is pretty impressive in this case. Okay, and so this would be a way that you could say, oh, these are length scales that I actually should be caring about or focusing on when I further study this problem. The other approach is now extending physics, or extending the theory with physics-constrained neural networks, right? So in this case, we take our uninterpretable neural network and now put in a physical bottleneck. And so this is saying every time I train my neural network, I'm going to constrain it by physics that I'm inputting as an experimentalist, right? And so here, the physics and you're only updating and you're minimizing your loss function with the rules of physics. And so here, what we put into this equation is, again, that physical model that we had used, which is the stressed elastic sheet, right? So we put in our trusted physical theory. And now, this is the scary part, and I won't say that we have it solved. You, as an experimentalist now, or your theorist, a scientist, you say, I'm going to allow these parameters to vary in a certain way, right? This is a recipe for complete overfitting and real BS on getting a result. But so this is where you really have to say, does this make sense? Does this not make sense, right? You're choosing where to fit parameters. In this case, what we did was we only allowed for a spatially varying adhesion field, because that made sense. Our theory did not account for a spatially varying adhesion field and also model parameters that were dependent on Zixin intensity, again, based on our grounded experimental knowledge, right? And so when we do this, now we can learn the parameters directly from the imaging data and really see how they, we can learn the parameters directly from our biophysical measurements. And so in this case, by using the physical bottleneck, we can learn where the adhesion field is, what the density, where the localization of the focal adhesions were, but also learn this stress-dependent Zixin intensity of how local stress is dependent on how Zixin intensity dependent on local stress, which again is learning that mechanochemical feedback. Okay. So, okay. So these are two different ways that now we can start to fill in this gap between the neural network models and the sort of physical, like interpretable models. But together, this is a really exciting new possibility in building models of living matter, right? Where you can now start to use AI approaches to really ground and learn the dynamics underlying the system, right? And it's a whole class of data-driven biophysical modeling where it still allows you to have interpretation and experimental use. Right, and I think one of the things that's super exciting for people who do imaging is that the images are highly data rich, right? The neural network training is easily achieved within an individual lab. And then all the statistical varying that usually as experimentalists we work really hard to engineer away becomes a feature. And if we go back to our paradigm, our 3M paradigm, right, this is really just blowing apart this paradigm. And I think it's possible to go back and forth between all these different ways of model building that we didn't have in the past that is going to allow us to now have a new ways of building interpretable physics of living systems. Okay, and with that, I just want to thank the people in my lab who did the work. The work I described today was really sort of a compilation of stuff over the past even 15 years. It was mostly led by Patrick Oakes and Cash and Shalaja and John Winkleman, and I mentioned Vincenzo Vitelli and his students who worked on the last stuff. The last thing I want to say is that the Institute for Theory in Mathematics and Biology, which is a Northwestern UChicago institute, and our Center for Living Systems, which is a Physics Frontier Center, are both doing open calls for postdoc fellowships. So if you're looking on the market for a postdoc, please write to us. The deadlines are on October 15th and November 1st. And thank you very much.
All right, thank you for the great talk. So we have time for questions. Let's see. Please raise your hand and then you got it, okay. And let's just wait until you get the microphone. And we got 2 mics, so somebody wants to stand in line.
That was a really beautiful and exciting talk. So how has this begun to shape your ideas for how you identify next best gaps to address? So, where this new approach of sort of taking the system apart allows you to identify missing factors or missing processes, a missing feedback loop, and then to prioritize which of those gaps is most important to address first.
Yeah, no, that's a good question. I think there's so much exciting work being done in this field. I think my approach, I'm pretty conservative, is I'm taking systems that I have a lot of intuition about just to do the methodology development. So for an example is you take a movie of dynamics of cells moving around. Could you learn the interaction forces in that movie? Could you learn the dynamics underlying the forward trajectories of the cells, right? It's not the most interesting biologic. I think there's like a bunch of different ways you could tackle this, right? You could just take the uninterpretable neural networks and just say, am I measuring something that's predictive of a field X? I think there's a lot of work just to be done on that without doing any interpretation as well. And that, I think, would help you know, is what you're measuring sufficient to make a prediction of something else? I mean, I think some of the most exciting areas of this, are examples where how much information is in phase contrast images of cells, right? How much do I actually have to measure to care about cell cycle or cell motility or differentiation, right? Could I just get that all from phase imaging of the nucleus? Something like that. There's like so many different ways depending on your inclinations that you could be asking these questions.
Yeah.
Yeah.
Yeah, fantastic as always, Margaret. So I really like the idea that the Zixin feedback is actually setting the mechanical properties not via the basic mechanics of the network, but via the feedback circuits itself. So maybe say a little bit more about how you think the properties of these feedback circuit actually set that. So if you don't just get rid of Zixin, but titrate Zixin, do you change the set point? Do you change the speed at which it actually gets to the set point? Can you understand a little bit more about how that feedback circuit sets the mechanics?
Yeah, I mean, I would love to work on that. So if you know anybody who wants to work on that in my group, that'd be great. Yeah, no, I think that's the question is like when you have a rate or a feedback loop that has a time scale or associated with it, if you tune that, how do you actually change the viscoelastic response, right? Yeah, that would be, and also, you know, I didn't mention it, but like that really dramatic difference between something that's mostly viscous and elastic is tuned by a whole collection of molecules that we actually now think that are undergoing in some kind of like liquid condensate together, right? So can you imagine some other mesoscale property of all those molecules working together that's actually setting the feedback that is also important? I mean, there's lots of, I think there's, I don't have the answer to that, but I think it's really exciting. Yeah.
Margaret, that was really insightful. Thank you. If I could ask two questions. First, the physics agnostic model building. I was with you, and then you said, and then we applied this Green's function layer. And as a developmental biologist, I'm wondering, could you just give a shred of insight for those of us who have never, like, what was that magic thing you pulled?
Electrons, right? You have two electrons, right? And then you know about electromagnetism. And the electromagnetism tells you how those two electrons are going to interact with each other. So that's giving you, the source is your electron, and the Green's function is your rules of electromagnetism. Does that make sense?
And the intuition that led to, like, this is the right type of equation to apply for this biological problem, what was that?
So the Green's functions, we didn't have to make that decision. We basically asked, what's the minimum number of expressions that we could use to make something that we were okay with?
Cool.
Because you could add as many functional forms as you want. So we just made it as minimal, but it's only math. I mean, like, math is useful, right? But it doesn't tell us electromagnetism, right? Like, it doesn't, it just gives us an idea of a length scale and objects and what the concentration of the objects are in terms of predicting what the response is.
And so that emerged.
Yeah, the judgment call came from the physical bottleneck where we had to decide how comfortable we are with, like, basically parameter fitting. And this is a really scary thing about the neural networks, is that you can really just overfit your data, like, and get really things that don't make any sense. So
and how far are we from using this kind of approach on, say, three-dimensional tissues?
Oh, it's, I mean, if there are people, we need really bright people working on this. There's a lot of work to be done. I was admitting to Lucy that, you know, this project was driven by these two really bright students who came from physics theory but were willing to learn some data science and computer science, right? And we need more people like you who are willing, and also experimentalists willing to dabble in it, because it's like I feel in this field like I felt going between physics and cell biology 20 years ago. It's like a whole new playing ground, and people need to be willing to get their hands dirty and try things that are just going to fail. There's a lot of issues. I'm happy to go on for hours about it. Yeah, these are not things that are out of the box tools yet. Yeah.
Hi. So the time evolution of some of the parameters in your physics bottleneck neural network, are these the important parameters from adaptive matter you were talking about? Okay.
Yeah, that's a great question. So I totally brushed under the rug, but that whole neural network approach wasn't doing any forward in time prediction. That was telling me in space, if I have difference of intensity fields, this is how your stresses are changing. So one of the things we're trying to do now is ask, how do I do that exact same problem and predict something future in time? So what are the transitions as something is like, how does that stress field or the intensity field vary sort of spatially and in time to allow for this smooth evolution, right, of parameters? Does that make sense? So we have not tackled forward time prediction yet, but that's what we're trying to do.
Hello. Thank you so much for the enlightening talk. My question is, what is the role in model building in the response to light as a function of wavelength? in the models, whether it's neural networks or optical tweezers. I didn't see anything about.
You need to, so here, when we're using light, we're using it to excite fluorescent proteins or fluorescent biosensors. And if the wavelength there is only to excite your fluorophores, if you're seeing any other effect of your wavelength dependence on your results, you need to redesign your experiment. Does that make sense? Like we try to use tools that do not impact our living system. And so if that becomes something that you see any differences, you just have to rethink how you're designing stuff. Does that, yeah.
All right. Cool. Well, let's thank Margaret again.
Cool, thanks.
Thank you so much. And